Hi guys, welcome back to my YouTube channel. I'm your tutor, Mr. Baraka, and today I want us to go through paper 2 so that we remind ourselves a few things as we prepare for Kcse Biology paper 2 2022. And you can see the first part that is there is what genetics. So there's always a question on genetics. And what do we expect from this topic? Most of the times, if you have a question from genetics in paper 2, the question will be dealing on what? Genetic crosses. So it is important to understand the components of a genetic cross because it is usually 4 marks. So you're supposed to get, those marks are supposed to be 3 marks. So how do you get the 4 marks? The first mark is what? The parental genotypes. You have to know the genotypes of the parents, and you write them correctly. And then in between the genotypes of the parents, there must be a capital X. Like the one that I have here. So in between the genotypes of parents, a capital X is the first mark. Then the second mark is for gametes. You have to draw circles that are complete. So if the circles are incomplete, you'll be penalized. So you draw complete circles of the same sizes to represent gametes. That is the second mark. The third mark is for fusion lines. You have to ensure that your lines do not hang. And I will show you. Lines do not fuse before touching the gametes, and the lines should not poke into the gamete. They don't get into the gametes. So if you have observed all that, then you have the third mark for fusion lines, and the fourth mark is the mark for the genotypes of the offspring. So you have 4 marks for a genetic cross. There's an example that I have given here so that you understand what I mean here. A cross between a carrier woman for albinism and a normal man. So if the woman is a carrier, it means the woman is heterozygous. So you have a dominant gene and a recessive gene, and then the man is normal, homozygous dominant, like that. So in other words, if you look at these genotypes of the parents, this genotype of the woman is correct, this genotype of the man is correct, so there's an X in between. If you show it like this, then you get the first mark there. And then we said from the genotypes of the parents, you can get what gametes. So can you see my circles are complete circles, complete circles like that. So you have this one coming in here. This is the gamete. This one is another gamete, another gamete, another gamete. So if the circles are complete, then you get what? The second mark for gametes. And then from there you come to fusion lines. You can see this one here. From here it comes all the way to this side like that, and then you have the genotype AA, the same thing like that. So you can see the fusion lines are just touching the gamete. They don't hang, they don't fuse before touching the gamete, they don't get into the gamete. So you get the third mark. And then the fourth mark is for what? The genotypes of the offsprings. You have a total of 4 marks. Put together. Now if you look at this one, you will find that there's another option here. You have the genotypes of the parents, they are there, but now the problem is that the X is missing. So because the X is missing here, it means then the mark for parental genotype goes. You miss that mark. This one is wrong. And then you come to the gametes. You can see the gametes are okay. They are circled completely. So this one will get the mark for gamete, but the mark for parental genotypes goes. And then if you look at this one here, look at the fusion lines. Can you see this one is poking into? So this one is wrong. Look at this one here. This one is hanging. It's not touching the gamete. This one is wrong. Look at these two. They're touching each other. They fuse before touching the gamete. This is wrong. So the mark for fusion line goes. So the only marks that this person can get is what? The mark for gametes and the mark of the genotypes of the offsprings. So it means you have 2 out of 4 instead of having 4 out of 4. So it is very important that you make sure that you follow those rules. And then when we say that circles are supposed to be complete, it means they should be like this, like what you have here or what you have here. If you have this scenario, it is wrong. If you have a circle that looks like that, this is wrong because you can see this circle is not complete, and therefore the mark for gamete will go if you have such a gamete. I hope you understand. Then the other part that you can get from the crosses that you are told to do what? To get either genotypic ratio or phenotypic ratio. For ratios, you give them in simplified forms. For example, if I use this Kcse here, you can see if you are to give the genotypic ratio of the offsprings, you can see there are two homozygous dominant similar like what you have here, and then you have two that are heterozygous like that. So you don't give it like this, 2AA is to 2AA like that. You have to simplify. So even if you write it like this, at the bottom here you have to show it is 1:1, so that you show it what simplify. So this one is correct if it's given like that. Or you can give it like this, 1AA is to 1AA like that, correct. Or you show the gametes up and then the numbers below here, so it is still what correct. Any of this is correct. The only problem is when you leave it like this, 2AA is to 2A like that. That one will be wrong because you have not simplified. I hope you have understood. Now there are some characteristics that are sex linked. So when you are dealing with characteristics that are sex linked, you must always show what? The sex chromosomes. They must be shown when you are showing the genotypes of the parents. If you omit the sex chromosomes, it means the genotypes are wrong, and therefore everything else becomes wrong apart from fusion lines. If they are correctly done, you will get 1 mark out of 4. So I want to show you, there's an example here that you have what? A hemophiliac man marries what? A carrier woman. So we get the genotypes of their offsprings. So you can see the woman is a carrier, and because the woman is a carrier, a female human being is XX. So you show the sex chromosomes and the genes, because the genes are carried by what? The sex chromosomes. So if the woman is a carrier, then the woman is having what? A dominant gene, capital H for normal clotting factors, and then the small h is for what? It's for hemophilia. So the woman is a carrier, so you can see XX and the genes are shown there. And then the man is a hemophiliac, so X and then a small h Y. So this gene is found on the X chromosome. The Y chromosome cannot carry the gene. And then you say these are the parental genotypes. Where you have the genotypes, what do? You put an X like that. You put an X like that, so you get the first mark for parental genotypes. The X must be there. You omit the X, you miss that mark. Then you come this side. We said you get gametes. You can see they are complete circles. You carry the chromosome and the gene together like that. Like that, you get what? A mark. And then fusion lines correctly done, you can see all of them. You get what? Third mark. And then genotypes with the offsprings, fourth mark. So you get a total of 4 marks from here. And then you can be told to get what? Probability of getting what? A hemophiliac son. If you are giving probability, you give it in simplified fractions or in percentage. So if you are looking for probability of getting a hemophiliac son, how many sons are here? There are only 2 boys. One is here, XY, and the other one here, XY. So there are 2 sons. But out of the 2 sons, one is suffering from hemophilia. So it is 1 out of the 2 sons, or 50%. So you can give it like that, or as a percentage. One of them is okay. And then sometimes you can be told to do what? You show the genotypes of the offsprings using a Punnett square or a checkerboard. Now if you are using a Punnett square, these are the components. You first show what? Parental genotypes with the X in between, and then you draw the square. You show everything that is involved in the square and you get the marks. Whether it is a genetic cross or a Punnett square, the marks remain the same. It's 4 marks. Now look at this one here. A Punnett square looks like this. If I was to use the similar cross of a woman who is a carrier for albinism and a man who is normal, then you see you start with what? Parental genotypes. So you show it, and X must be there, first mark. Then after that, you draw the square. So what do? You draw the square like that, divide it into 2 horizontally, and then into 2 vertically like that. And then you go to the first square, and you divide the first small square diagonally like this. And you can see like that. So that this one here will be for this parent, and this one here will be for this parent. So you can see, what do you write here? You write what? The genotypes of the parents. So you can write AA like that, and then you have it like this. The other side, this one comes this side, AA like that. And then this one and this one gives you AA. This one and this one gives you AA. This one and this one gives you that. And then this one and this one gives you that. So the first mark is here. The second mark comes here when you have split this one diagonally. They're supposed to be, and then the third mark is this plus here. And you see this plus here is like fusion lines. So whatever you see here from here to here and here to here, this is a fusion line. So you have to do it properly. So this line must come and touch here. This one must come and touch here. So you don't go beyond. You don't go beyond, because if you go beyond, then it is wrong. You will be penalized. So like that. So if the plus is okay, fusion line, you get a mark, and then the last mark is what? Mark for megametres. So you have 1, 2, 3, 4, just like a normal genetic cross. Now, when are you penalized for a Punnett square? Look at this one here. I have this one and this one. These are parental genotypes, but now the X is missing, so the mark will go. And then I come here. Look at this part. Can you see this small square? This person has not divided this small square diagonal like this. So it means it's like having one parent. That whatever you have here, this side and this side is the same thing, the same parent, which is wrong. So you must divide this one like that. So because this person has not divided this small square diagonal like that, then the mark for this goes. There's no mark here. You lose the second mark. And then you come here. Can you see this plus? You can see this person has extended this side. The other side also extended. So if you extend here, it's wrong. You extend this side, wrong. So this mark will go. The only mark this person will get is what? The mark for the genotypes of so you'll have 1 out of 4 instead of having what? 4 out of 4. I hope you have understood how you draw what? A Punnett square, so that you get maximum what? Points for that question. So as I conclude, you realize that I have not written what? Parental phenotypes up here. I have not written anything on this side because whatever you write here and the parental phenotype, they have no marks. But now if you choose to write, you make sure that they are correct, because if you write wrong phenotypes and the genotypes are correct, then the wrong phenotypes will cancel the correct word, genotypes. That is why it is better if you don't write anything. You just write it the way I have written here, without writing anything on the left hand side, or without showing what? The phenotypes of the parents, because they have no marks. Now the other part that we expect to get a question is what? On experimental biology. What do I mean here? It means you are given a question that is involving what? An experiment, and it is you to understand what is happening in the experiment. Sometimes you are even told to give what? The aim of the experiment, and then you explain the observation that are there in that experiment. So it is you to understand. And most of the times the experiments are based on what? They might be based on the photosynthesis process. They may be based on what? Respiration, like what? Anaerobic respiration. They might be based on what? How urine is formed. They might be based on what? Ecology, especially on what? Estimation of population, capture method. They might be based on what? Cell physiology, mainly osmosis or diffusion. They might be based on tropism, phototropism, geotropism, and thigmotropism. They might also be based on what? Gaseous exchange, like if you are given an experiment on the model on mechanism of what? Breathing in a mammal, you have to understand the experiment, and therefore you'll be able to answer the question that follow after the experiment. So it is very important that you go through these areas and you understand. You look at all the experiments that are there, especially using KLB. Experiments that are there based on these areas, you will be safe, because you are likely to get a question on a certain experiment based on these areas that are here. Then the other area is what? Support and movement. You are most likely to get what? A question that is based on what? On bones. And when I talk of bones, I talk of what? Bones of what? Of the appendicular skeleton. And when you talk of these bones, they mainly involve what? The scapula. Maybe you're given the scapula, you identify the bone, the region found, and adaptations of the bone. So scapula, we have humerus here, we have ulna and radius. We also have what? We have pelvic girdle, we have femur, we have tibia and fibula. All those bones, you have to remember them. Then when we talk of vertebrae, these are bones of what? The vertebral column. So bones around the neck region, atlas axis. You have to remember those bones, the way they look like, even the structure of a typical cervical vertebra. The adaptation of those bones is very important. Thoracic vertebra, how you can identify it if you are given a photograph. Adaptation of thoracic vertebra to its function. Also lumbar vertebra, the same thing, adaptations. The sacrum adaptations. You must be able to understand all those vertebrae because you are most likely to get a question from there. And then for joints, the main joints are ball and socket and hinge joint. And you said, where do you find this ball? If you are to get these joints, you will find that you are either given what the shoulder joint, which is ball and socket, or you are given what the hip joint, which is ball and socket. For hinge joint, is the elbow joint is a hinge joint, or the knee joint is a hinge joint. So you're supposed to know that. It's very important. You're likely to get a question from there. And then we go to classification. For classification, you look at what? Mostly in plants, divisions, bryophyta and pteridophyta, very important. You have to know how bryophytes look like and the characteristics. The same thing with what? Pteridophytes. And then classes in plants is mainly what? Dicotyledonei and monocotyledonei. You have to be able to understand those classes. For animals, phylum is the phylum Arthropoda, which is very, very common. You can also get what? Phylum Chordata, but Arthropoda is very common. Classes of animals that are very common. One of them is what? Crustacea. You identify, you are given the organism, you identify the class, and then you give what? Some characteristics. It's very important. Classes Insecta and Arachnida. Classes Amphibia and Reptilia. Class Aves. For Chordata, class Aves, very important. For birds. Class Mammalia, for mammals, very important. And we also have what? Class Diplopoda and Chilopoda for Arthropoda. So these ones are these classes are very common. So you might be given an organism from here, and then you are told to classify the organism, giving characteristics and all that. So you must be able to go through and have content on these areas. It will be very important. Now for section B of paper 2, you go to graph work. Now I have given you, there are 4 topics that are most likely to give us a graph question. Ecology, excretion, and response and nutrition. These 4 topics, they are likely to give us what? A graph question. So it is you to go through any revision book that is having what? Graph questions on ecology, excretion, response, and nutrition. You remind yourselves those areas. You are most likely to meet a question from there. Now, the components of what? Of a correct biological graph. Drawing of a graph in biology is supposed to be 3 marks. So you have to know how do you get the marks. If you know how to get the marks, then it will be very good for you, because you are likely to score all the marks from that graph. Now the first thing is this, that for a biological graph, you must always label both axes x and y, and the question directs you, because maybe you're told to draw a graph of this against that. So the first factor, you label it on your y-axis, the second factor on your x-axis. So when you label both axes, you have your 2 marks free. And then after that, now you go to the scale. So the scale is correct if you have what? Origin or origins. So you can have 1 origin for both, or you can have 2 origins. Origin for x, origin for y. And then the scale must have an origin or origins. It must be consistent. It must be workable. You must ensure that the curve that you are drawing, the scale must help you have a curve that is occupying more than half of the paper that is provided. So it must be a big curve. It must be big curves, not small, tiny curves. So you have to use most of the space that is provided in that graph paper. And then the scale must accommodate toward the upper limit of the data provided. So if the highest value in your data is 100, then it means your scale must go up to 100 or beyond 100. Now, if the origin is missing, it means the scales are wrong. If the scale is inconsistent, still wrong. If you cannot use your scale to plot the points, it's still wrong. If the scale is making you to draw very tiny curves, the scale is still wrong. If the scale is having is not accommodating the highest value of the data provided, you are still wrong. So make sure that you follow all these to earn 2 marks for the scale x and y. And then plotting, you always plot using crosses. Don't use dots, because sometimes when you draw the curve, the dots cannot be seen. So use crosses to earn to earn marks for your plotting. And ensure that you plot all the values that are provided in that table. Don't leave any value in the table. Make sure that all the values in the table are plotted correctly. And then for the curves, make sure that you draw smooth curves. You use your free hand. There is nowhere you will be joining the points using your ruler. It is always using your free hand. And the curve must be a smooth curve. And you draw it once, smooth curve. Then if you have more than one curve that is drawn, you have to identify the curves, so that you show us which curve represents what. So that is what we mean by identification. You identify the curves that you have drawn, especially if you have more than one curve being drawn, you have to identify the curve correctly. So you can see labeling of axes is 2 marks. The scale is also 2 marks, so there are 4 marks. Plotting, if you have 2 curves being drawn, you will plot for 2. That means you have now 2, so you have 6. And then if you have 2 curves, each curve is a half, so it gives you 1 mark. And then identification of the 2 curves, each a half, you have a total of 7 marks. So you have 1, 2, 3, 4, 5, 6, 7, and then this one for the derivation is a half times 2, so you have another mark. So you have at least 8 marks actually. So you have 1, 2, 3, 4, 5, 6, 7, 8. Those are 8 free marks if you are drawing 2 curves. Even without attempting anything after the graph, you have already 8 marks. Then if you come this side, I have tried to show you the origins for the scale. For example, if your x-axis has the lowest value of 0, then look at the way I have done here. You always extend the origin. You extend this side and this side. So the value that you put here is the origin for x. The value that you put here is the origin for y. So if maybe the lowest value for y is 100, then you don't have to start from 0. Use 100 as your origin. You put it like that: 100 origin for y, 0 origin for x. If the data is having 0 as origin for both, you can put your 0 here and 0 here. And you see I've extended like that. So this 0 here origin for x, 0 here origin for y. Like this one is still correct. Instead of putting like this, you can put like this. This 0 is origin for x, this 0 is origin for y. Then this one is very correct also. You just put a 0 here at the center. So this 0 here is for origin for both x and y. So x 0, y 0. So the origin is okay. Yeah. So these are the correct ways of showing the origins. Now if you look at this one, you can see I've put an x. This one is wrong. Why? Because this 0 is the origin for x. But now if I was to draw an imaginary line here like that, you will find that this 0 is for x, y has no origin. And if y has no origin, it means the scale for y-axis is wrong. Whatever you plot is wrong. Whatever you draw as curve is wrong. So you must make sure that you must have origins for both axes. This one is also wrong, because if I draw an imaginary line here, this 0 is for y, x has no origin. So this scale here is wrong. Plotting and curve, everything becomes wrong. Look at this one here. This person has not extended, so it's very dangerous. Because if you put the 0 here, if I draw an imaginary line, this 0 is for x, y has no origin. So this scale is wrong, plotting is wrong, curve is wrong. If you come this side, the same thing. If I draw an imaginary line, this 0 is for y, the x does not have an origin. So x scale is wrong, plotting is wrong, curve is wrong. So this one becomes very dangerous. So you must make sure that you always extend the origin, so that you show the origins properly the way I've showed you up there. Then we come to essays, the last part, essay writing. I've written here something. You always choose the essay that is easier for you. And how does it become easier for you? If you have more points, because there are 2 essay questions, question 7 and question 8. So you will choose the question that you have more points, and you tackle that question. And essay questions that are based on processes, you must be very careful, because if it's a process, you have to follow the steps carefully, because if you omit any step, then it means marking stops there. That is why you will only choose a question on the process if you are very sure of all the steps involved, and therefore you can do it properly without omitting any step. But the best essays to tackle are essays that are based on adaptations of organs. These ones are the easiest, because now every time you name any part of the structure of that organ, the name of the structure is a mark, and then now the other mark is what? On the adaptation to function. So correct adaptation and function, a mark. The name of the structure of the organ, correct name, a mark. So you have 2 marks for each statement that you make. So you can see this one is easier, and there is no format. You can start with any part of that organ, and you still score what? The marks. What is important that you have to ensure that there's good flow of ideas or points as you are writing your essay question, and you make sure that your handwriting is legible, can be read. Whatever you're writing can be read, and you must be neat. That is basically what is very important. Now I've given you an example here so that you see how easy it is to answer what? And a question based on adaptation of organs. You can see the question here says what? Describe adaptation of parts of mammalian eye to their functions. So you can see the first part is cornea. So I've just given cornea. If you write cornea, correct spellings, you can see semicolon means mark. So the tick is here. You get a tick. If you say cornea, correct spellings, tick. And then adaptation function. Adaptation is curved. Function to refract light. So you can see another mark here. So you have 2 marks for them. Cornea, the name of the structure, tick. Curved, adaptation to refract light, function. Correct? Both of them correct, tick. If one of them is wrong, you don't get a mark. But both of them correct, get a mark. Then the other part is what? Conjunctiva, correct spellings, tick, mark. Transparent, to allow light to pass through, another mark. Look at this one here, pupil, a mark. An opening or a hole that allows what? Light to pass through, another mark. So that is how easy questions on adaptations of what? Organs are. So you're supposed to, if you see such questions, you must always go for such questions, because you will earn a lot of marks without struggling. Now, from my analysis, I have realized that these 4 topics are the ones that are likely to give us what? To give us essay questions for 2022. So I have indicated them here. You can see ecology, excretion, response, and nutrition. And what do you expect from ecology? You can see abiotic factors, how these factors are influence what? Distribution of organisms in ecosystem. Then nitrogen cycle, and then plant adaptation. This is what? Adaptation of xerophiles, halophiles, hydrophiles, mesophiles to their habitats. And then the other one is pollution, water, air, and soil pollution, the causes, effects, and control. So basically, if you get an essay question from ecology, these are the areas that you're likely to get. And then for excretion, you're likely to get what? A skin. And for the skin, adaptation of what? The parts of the skin to their function, or role of the skin in what? Thermoregulation. And then kidney, and this one is basically what? How urine is formed. Urine formation, 20 marks. This is a process. And then liver is what? Homeostatic functions of the liver, very important. And then role of hormones in homeostasis, and the main hormones are what? Insulin, glucagon. There's another one that is aldosterone. There's another one that is what? Antidiuretic hormone. All of them, those 4 hormones, you must know how they are involved in homeostasis. And then excretion in plants, mainly what? The economic importance of plant excretory products. So you have to know that. That is what you expect from excretion. And then from response, it is mainly in what? In animals, you get what? Reflex actions, simple and conditioned reflex action, how they happen. You must be aware of that. And then if it's organs, it will be eye or ear. This is what? Adaptation of parts of the eye to to their functions, or parts of the ear to their functions. You must be aware of that, very important. And then for plants, it is tropism, phototropism, geotropism, and thigmotropism. You must understand what happens in all these tropisms, the role of auxins in each of these tropisms. And then for nutrition, it is a leaf adaptation. That means adaptation of what? Of a green leaf to its photosynthetic function. You must know all the adaptation. And then process of digestion, you have to know what happens. How do you digest proteins? How you digest what? Digest lipids, how you digest carbohydrates, very important. And then adaptation of ileum to its function, or you can be told adaptation of small intestines to its function. You must be aware of that. And then the last part of nutrition is photosynthesis and factors affecting what? Photosynthesis. So if you're able to go through this, you increase what? The chances of scoring highly in your essay what? Question. Otherwise, I believe that this information has helped you as you prepare for your Kcse Biology Paper 2 2022. I wish you all the best, and let's meet next time.